Okay, so welcome to this next video in which we are discussing the adaptive immune system and immunosuppressants. Okay, so in this video what we're going to talk about is another drug which stops proliferation within B and T lymphocytes and is to an extent selective for B and T lymphocytes and will only stop cellular division in those cells, and therefore will have fewer side effects than a drug which just has an anti-proliferative effect in all cells, such as cyclophosphamide. Okay, so this is the drug azathioprine. Okay, so this drug is again going to stop the synthesis of guanine nucleotides of both the RNA type and the DNA type, but it's also going to do something slightly extra. It's also going to stop the synthesis of adenine nucleotides as well. Now, adenine and guanine have a very similar structure. They're both purine organic bases. So, azathioprine stops the synthesis of the purine organic bases, is what you would say. Okay, and if you stop the synthesis of adenine and guanine nucleotides that are incorporated both into DNA and RNA, then you're going to stop DNA replication as well as stopping protein synthesis, and those are the two things that you really need in order to divide, and hence if those aren't working properly, the cell is not going to divide. And the reason that they're selective for T and B lymphocytes, the azathioprine is selective for T and B lymphocytes, is that other cells will have other mechanisms for synthesizing these purines, which will allow them to get past this blockade of this synthesis, whereas the T and B lymphocytes lack these mechanisms and therefore won't be able to get around it. Okay, so what does azathioprine do? Okay, well, this is the big name of the enzyme it inhibits. So it's going to inhibit an enzyme known as amidophosphorybosyl transferase. So amidophosphorybosyl transferase. Okay, so what does this enzyme, amidophosphorybosyl transferase, actually do? Well, basically, it catalyzes the conversion of a molecule known as alpha-phosphoribosyl pyrophosphate into what's known as 5-beta-phosphoribosylamine. Okay, so let's see the structures of these molecules and then try and understand why this is so important for the synthesis of adenine and guanine organic bases, or rather the adenine and guanine nucleotides from scratch. Okay, so we'll start with this molecule which is known as alpha-phosphoribosyl pyrophosphate. So alpha-phosphoribosyl pyrophosphate, and I promise you the name is far scarier than the actual molecule. The molecule is just ribose with a few phosphate groups stuck onto it. Okay, so alpha-phosphoribosyl pyrophosphate is often abbreviated to alpha and then little dash in between, PR for phosphoribosyl, and then PP for pyrophosphate. So let me show you the structure of this. So you'll have your ribose here, okay, which remember is this 5-membered ring where one of the members is an oxygen and the 4 others are carbons. Now, let's show the optical isomerism for once, okay? So, each of these carbons is going to have an important group coming off it, and then also a hydrogen atom coming off it. So, basically, let's discuss whether the important group comes out of the page towards us, or the hydrogen goes away, or whether the important group goes away into the board or into the page away from us. Okay, so this actually is important because there are other molecules which have slightly different optical isomerism to ribose, which are considered different molecules. Okay, so we'll start off with the 5th carbon here. So the 5th carbon comes out of the page towards us basically, and then the hydrogen off this 4th carbon back here goes into the page away from us. Okay, so here is the 5th carbon coming out of the page towards us. and then it's going to have an alcohol group coming off it here with its phosphate group here. Okay, so this is the phosphoribosyl portion. This is the phosphate group of ribose. Okay, so that's the optical isomerism of the 5th carbon up here. It comes out towards us, whereas the hydrogen that is coming off this 4th carbon here is going into the page away from us. Now let's consider this 3rd carbon. This 3rd carbon has an alcohol group coming off it which goes into the page away from us, and the hydrogen that comes off this 3rd carbon will come out of the page towards us. Then the 2nd carbon, again, the alcohol group goes into the page whilst the hydrogen comes out of the page towards us. And now this position here is mixed, basically. The important group can either go into the page away from us, and that would be called the alpha isomer, or it can come out of the page towards us, and that would be called the beta isomer. Now we're talking about alpha phosphoribosyl pyrophosphate, so this is the alpha isomer, so the important group is going to go away from us into the page, and the hydrogen will come out at us. Okay, so what is the important group? Well, this is this pyrophosphate. So you would normally have an alcohol group here on a normal pure ribose molecule. However, we're talking about ribosyl pyrophosphate. So instead what you're going to have is 2 phosphate groups which are linked onto this. Okay, so here's one phosphate group, and then here's the next. Okay, so this is the pyrophosphate group of this molecule. So this is the pyrophosphate bit here, ribosyl is this bit in the middle, and then the phospho here denotes the fact that you have a phosphate group stuck off the 5th carbon up here. So this is alpha phosphoribosyl pyrophosphate. Okay, now this enzyme, amidophosphoribosyl transferase, is going to convert this enzyme into something known as 5-beta, okay? So it's going to be the beta isomer rather than the alpha isomer. And then it's phosphoribosylamine. Okay. So, phosphoribosylamine. Okay, so let me show you the structure of this molecule, and then I'll talk you through how the enzyme amidophosphoribosyl transferase is going to catalyze this reaction. So, most of the molecule remains exactly the same. So let's draw it out again. Okay, so here is the 5-membered ring here, and then off this 3rd carbon you have an alcohol group going into the page away from us. Off the 2nd carbon you have an alcohol group going into the page away from us. Okay, then off this 4th carbon the important group comes out of the page towards us, and this is the 5th carbon of the ribose sugar. And then off the 5th carbon you have this phosphate group still. Okay, so I'll put this here. Okay, and then it will have its alcohol group over here, and then oxygen down here. Okay, but now the change is that firstly the important group of this 1st carbon is going to come out of the page towards us because we're talking about the beta isomer here. And then you have an amine group there, rather than an alcohol group, which makes this ribosylamine. Okay, now we'll continue this discussion in the next video.